Hi, I'm Julianne, co-author of a novel approach to Huntington's disease. It employs a physics-based method that naturally connects the usually separate disciplines of cardiology, pulmonology, vascular physiology, and neurology. We discuss how to quantify the health of the so-called neurovascular unit, which we use to characterize Huntington's disease. To begin with, let's go through the cardiovascular system. The cardiovascular system consists of the heart, the lungs, the blood vessels, and the blood. Blood flows continuously around the closed systems of vessels. It delivers oxygen and nutrients to cells and removes their waste. This flow must be finely regulated, involving many different oscillatory processes. To show their effects, we consider the journey of red blood cells to the capillary bed. The main function of a red blood cell is to transport oxygen to be used in the production of useful energy in the cells. It must first be collected from the lungs by deoxygenated red blood cells. The lungs inflate and deflate at around 15 times per minute. Once the red blood cells have collected oxygen from the lungs, the heart pumps them around the rest of the body. The heart beats around 60 times per minute. The heart and lungs are examples of biological oscillators that give rise to rhythms. The velocity of flow is regulated by many local oscillatory processes in addition to the heart and respiratory rates. In the arteries and arterioles, rings of smooth muscle cells continuously expand and contract to adjust the flow. Vasomotion involves oscillations in the diameter of the vessel wall, which usually occur around 6 times per minute and can be observed in blood flow signals. The nervous system can also influence blood flow. Nerves surrounding the vessels can signal to the muscle cells, causing even more vasomotion. Further, vessel walls are aligned with a thin sheet of endothelial cells. One of their functions is the regulation of blood flow by rhythmic release of substances that cause vasomotion. This is one of the slowest rhythms in the cardiovascular system. In the capillaries, the smallest blood vessels, nutrients and oxygens are rhythmically delivered to the cells. The capillaries are very narrow, deforming the red blood cells and thus helping to release the oxygen. Biological oscillations do not stop once the oxygen is delivered. They can also be found on even smaller scales. Within individual cells, oxygen is used in energy metabolism to rhythmically produce the energy-carrying molecules known as ATP. The further the blood flows from the lungs and heart, the more oscillations become involved in the regulation of blood flow. So what does this mean for the brain? The neurovascular unit consists of the microvasculature, neurons, and astrocytes that connects them, and it's responsible for the regulation of blood flow in the brain. The mutant huntingtin protein has been found in all major parts of the neurovascular unit. Still, a non-invasive assessment of the functioning of the neurovascular unit has not been done in Huntington's disease patients. We combined non-invasive measurements of brain oxygenation via functional near-infrared spectroscopy, brain electrical activity via EEG, and cardiorespiratory function via a respiration belt and ECG, with novel mathematical techniques developed to study biological oscillations. These techniques aim to characterize the time-dependent finite time dynamics of biological systems and include the wavelet phase coherence and methods available in the toolbox MODA. Our results show a reduced efficiency of the neurovascular unit in Huntington's disease, both in the presymptomatic and symptomatic stages of the disease, as the neurovascular phase coherence in the interval around 0.1 Hz is significantly reduced in both disease groups. The vasculature is already significantly impaired in the presymptomatic stage of the disease, shown by lower power of oxygenation oscillations compared to controls. The spatial phase coherence of the oxygenation oscillations is lower in the symptomatic Huntington's disease group compared to controls, which indicates vascular remodeling, which is most likely a compensatory mechanism. In contrast, the EEG power results and decline in alpha and gamma coherence indicate a gradual deterioration of neuronal activity. The observation of altered dynamics paves the way for a simple method to monitor the progression of Huntington's disease non-invasively. We also hope that our study will stimulate new treatments of Huntington's disease targeting the vasculature and brain metabolism.